Logan? Logan?
Yep, looks good.
Great, thank you. All right, good morning, everyone. Good afternoon, wherever you are. Thanks for joining us for the FIND today. My name is Lauren Hayes. I'm coordinating one of the co-organizers of the FIND, and today I'm your host. I'm really excited about today's seminar because we have a unique, a different structure today with student presentations. And before I introduce the three presenters for today, I just want to make a couple of quick announcements. So I'm going to share my screen here. Sandra, can you see that okay? Just give me a thumbs.
Yes.
Thank you. So first off, I want to thank last week's speaker, Joan Silk, for a fascinating seminar on primate parenting, and announce that next week we have Aura Raiolo from I'm forgetting where her university is, but she's going to be talking about the transmission of gut microbiota and how that is associated with social networks in wild mice. That should be a really fascinating seminar. And I want to just, since the theme of today is focusing on student work, I wanted to remind everyone and share with new people here that are joining us. And by the way, if you're new to the FIND, thanks for being here and welcome. We're happy that you're with us. We are promoting educational activities as much as we can through the FIND. We have a number of resources on our webpage. If you go to our webpage, Frontiers in Social Evolution Seminar, we have a number of links that you can go to get information or resources that may be useful to you, including teaching slides. And we also have, a reading list that you can go to if you want your students reading papers in your classes. And then some of us have even published a paper on using remote seminars in teaching in animal behavior to give you some ideas to take to your classes. We also have been promoting other activities to promote education at various levels. Eduardo led a workshop on scientific and statistical inference, making us think differently about how we use stats and interpret our data. We also had a long-term panel discussion on long-term studies a couple years ago. And something exciting that we're planning for the fall FIND is a panel on behavior and conservation. The idea that I think a lot of behavioral ecologists like me think, oh, I can generate data that conservation people can use, but it turns out it's a little more complex than that. And so we're going to have a couple of people come in to talk about that issue and engage in discussion with the community. We've also been having some students produce quick finds. These are short videos that are posted on our YouTube page. Here are just some screenshots of 3 that are posted. What they do is the students go in, they watch one of the FIND videos that intrigues them, and then they make a 5 to 7 minute video about that. And today I just received 3 new ones for my behavioral ecology class. And what was exciting about this is the graduate students in my behavioral ecology class, there are 3 of them, they collaborated with undergraduates in Adriana Maldonado Chaparro's classes in Colombia, and they produced quick finds together. And those soon will be posted on the webpage for everyone to see and watch if you're interested. It's a great activity if you want your students thinking about the FIND and communicating in a concise way. And I think the students benefit from it. And the theme of today is supporting students and junior scientists. And today's FIND, which we have 3 people joining us, was motivated by something that Karsten led back in the fall of 2022, where 3 African students gave really fascinating talks about their PhD research. And I think this motivated me to think about my connections in South America. And that's what we're working on today, is we have 3 students from Latin American countries giving talks. And the structure of the seminar today is a little different than usual. Because we have 3 talks, we'll have each student will give a 15-ish minute talk, and then we'll have some time for you to ask questions about that specific talk. And then We'll transition to the next, and then after the third talk, then we'll have that traditional time after the talk where the whole community can ask questions about anything that was discussed during those seminars. And so today we're going to have 3 talks, one by Sandra Smith Aguilar, Nelson Galvis, and Cecilia Carrizo. And at this point, I'll introduce each speaker independently. So I'm going to start with Sandra, and then she'll give her seminar. Then I'll introduce Nelson, and he'll give a seminar, and then I'll introduce Selena, and she'll give her seminar. And so to start with Sandra Smith Aguilar, Sandra did her bachelor undergrad degree at the National Autonomous University of Mexico. She then earned a master's degree at the Institute of Marine Science and Limnology at that university. And she's currently a postdoc working in the National Autonomous University of Mexico as well as the Institute for Research in Applied Mathematics. Sandra's generated some nice publications already in her career. Her papers have been published in journals like Primates, Frontiers, and PLoS One. Themes range from looking at socio-spatial structure in spider monkeys to promoting more computational approaches to understanding fission and fusion dynamics, and looking at multidimensional natures of social structure, focusing in spider monkeys. Sandra has indicated that, she's in the second year of her project studying social structures in these spider monkeys and is interested in looking for a postdoc in 2024. And, her interests are not just in spider monkeys, she's also interested in other mammals such as cetaceans. But I think the major theme of her work is using tools like social networks to understand social structure. And today, Sandra is going to be giving us a talk on her research on the spider monkeys. The title of her talk is what can multiplex networks tell us about the social dynamics of spider monkeys? And with that, I'm going to stop sharing my screen and turn the talk over to Sandra.
Thank you very much, Lauren. And I'm going to share my screen. Let's see. Let's just make sure that we get. Okay, so does that look good?
Yes, it does.
Okay, thank you. So hello again to everyone. Thanks for being here, and thank you to the Fine Seminar for organizing this event. And thanks to Filippo Aureli, particularly because he's the one who suggested I try to participate here, and here I am. So thank you very much, Filippo. And as Lauren said, and as we were discussing before this whole thing officially started in video and all, I really appreciate the existence of the Fine Seminar in this space, because I think it gives us a chance to present present things and plug into the brains of all of you nice people who are here today and get ideas and discuss them in a scenario which is a little bit different than from like a scientific meeting or something where you're more trying to present something finished. So that's what's going to happen on my part. Today, I'm bringing to you work that ongoing work and things, ideas that we're developing. And any insights that you get and you want to share with me, I would love to hear them. So let's go. So as Lauren said, I'm going to talk to you about what can multiplex networks tell us about the social dynamics of spider monkeys. And this is framed, let me wait just a second. Next slide. Yeah, okay, so this work is part of a bigger project which involves several institutions in Mexico, including the UNAM, and it's a project called Understanding the Social Structures Through a Complex Systems Approach. Emergence of dynamic flexible properties and multiplex social networks. So my postdoc is participating in this project, which is led by Gabriel Ramos-Fernandez, Denis Boyer, and Filippo Aureli, two of whom are here today, fortunately. And but this, I mean, they had the project. It includes a lot of people, some of which are here right now, and several institutions. So I'm lucky enough to get a chance to collaborate with all these nice minds here. So there's traces of their presence and their contributions to the ideas that I'm sharing here today, and many other people who have helped with data collection. But one of the central interests of this big project, of course, is looking at this interplay and between what happens, the decisions of individuals who live in a social group, and the patterns that result from those decisions in terms of interactions, giving rise to social structure, and how that feeds back to the decision-making process of the individuals and their interactions. So I really like this paper, the paper by Mauricio Cantor and collaborators, where they present this figure, and they highlight the importance of trying to look at these things and the loop that I'm showing here all together, because which is just recently getting more and more attention as a whole, let's say. It's not like a lot of people for decades haven't been looking at different parts of what you can see in this cycle, but what they're trying to call their attention to is that we have to kind of find ways, or we're trying to find ways to look at it together in order to get better understanding of what's going on in terms of the social patterns that emerge from the interactions and how they can feed back to what's going on with the individuals. So project's more or less about that. And I'm going to focus a little more. This is going to be more kind of a methodological talk, because what I'm doing for a large extent of my postdoc is trying to explore how well some tools, particularly multiplex networks, can help us in this mission to try to understand social structure and its properties. So for those who are not familiar with multiplex networks, they are a type of multi-layer networks. And when I say multi-layer networks, I mean networks where you can have several different types of links or interactions, let's say, for example, in the same frame. Like to the left here, I'm representing three layers, three networks, each of which has nodes with different colors, trying to represent different types of interactions. But on the far left, we can see that these layers or these networks are not connected between each other. Whereas in the central figure here, you can see these dashed lines connecting individuals, the same node, throughout different layers. So that's what makes a multiplex network, where having connections between an individual and its own self in other layers. So and it's a little bit different from a broader interconnected network frame, where you can have, rather than having connections between layers between the same individual or same node, you have connections. For example, if the top could be birds and the middle could be a tree, so you have the two birds connected to a same tree here. So we have these different types of multi-layer networks, and they're all very interesting. But for now, this talk, I'm just going to focus on this type of network, multi-layer networks, the multiplex. So as I was saying, usually, or it's very frequent, at least in animal behavior studies, to use these layers to represent different types of interactions among a group of individuals, animals. For example, here we have a study on mandrills, where each layer represents three types of interactions, here we have to the left agonism, proximity, and grooming. Among the same set of individuals, which are located in the same position in each layer. So this would make a multiplex network here, for example. Although this is perhaps the most common way that multiplex networks have begun to be used in animal behavior, another potential use or way to work with multiplex networks that has been proposed in several papers is considering each layer as a slice of time for an interaction, for example. So here, we can imagine this is 4 instances or 4 periods for the same interaction, representing the same interaction at 4 time points, different time points, no? So in this sense, we would have a temporal multiplex where you have the same individuals through time with the same types of interactions, showing different snapshots of what's going on. So it's different to a dynamic temporal network where you have the continuous presence of time as it goes. You still have snapshots of what's going on here, but you can represent several snapshots in one structure. And if you want to make it a little bit more complex, you can have several interaction layers. Like here, you have a green layer and a blue layer to the right, I'm sorry, which is a multiplex. And then you can have that same multiplex and several time periods. That would also be a temporal multiplex with more than one interaction type in itself. However, to keep things simple for now, I'm just going to talk about this type of multiplex where we have one same interaction, which is going to be associations, and how they display through time. So with this brief introduction on the methods, what I was trying to do and what I want to talk about is we were wondering, can we use temporal multiplex networks like the ones I just showed to learn something about the social dynamics of spider monkeys? And this type of idea has been worked on or used before. Particularly, I want to point out this paper, which is actually the one that triggered my curiosity to see if this type of framework could be used for spider monkeys in a very different way. But here, David Fisher and Noah Pinter-Woolman did a study on social spider where they had a certain type of interaction through different time periods to look at social stability. So that's more or less what I drew from this, using what an analysis proposed by De Domenico, which is called the reducibility analysis, which basically it's trying to help when in the multiplex framework or multi-layer framework, trying to help us decide how much redundance you have in a multiplex structure. So we don't want to have a lot of redundancy there because, I mean, having one layer is complex enough. Having 5 layers makes it even more complicated and difficult to interpret. So we are interested in trying to keep things as simple as possible. So having unnecessary layers because they're redundant is pointless. So that's what gives rise to this proposal of our analysis, which tries to reduce this, try to look which is the configuration of your multiplex which reduces redundancy the most, right? So for instance, for example, here, if you in this multiplex network, you have here on the top left, we have they're representing redundancy with the same colors, trying to indicate that all the orange layers are pretty much the same, whereas the yellow layers are pretty much the same, and the green layer is the same thing. So it would probably be better, or from this example, what they're trying to show is it would be better to just have 3 layers rather than have 10 layers as shown. And they developed the analysis through a measure based on the Shannon-Johnson distance to try to see which layers are most similar and how similar, I mean, how much information you keep or how different your multiplex structure is from a fully aggregated network by having a certain number of layers and certain layers aggregated or not. So this is a kind of way to see that, to detect redundancy. So here on the top, you have this little curve, blue curve here showing that with this cost function, which what it does is try to represent how much information. So the higher up on the y-axis you're here, is you're having more, it indicates that you have more information than the equivalent aggregate network, which is represented to the right here in the gray layer, where you just have one layer where you sandwiched everything into one, right? So in the example here, the highest point on this curve corresponds to a 3-layer configuration of the network on the left. So that would be the optimal configuration and less redundant in this case. So in the context of temporal framework, high stability or this high redundancy could also be read or interpreted as high stability. That is, if you have many layers that are very, very similar among each other through time, it kind of suggests that things are not changing too much. So that's more or less what I plugged into for what I'm going to present next. Keeping that in mind, I'm trying to see, or the thing I'm trying to do, we're trying to do here is to describe the temporal dynamics of associations in spider monkeys using this temporal multiplex framework and the reducibility analysis as the one I just explained. So to do this, I'm looking at data from a spider monkey group living in Yucatan, the Yucatan Peninsula in Mexico. This is southern Mexico. This is the Yucatan Peninsula in a small protected area called the Tochmash-Yetelco. And here we studied, or well, the data that I'm presenting is based on one wild habituated group of Jeffrey spider monkeys, which was studied continuously from 1997 till 2020. This is 24 years of monitoring of subgroup data, which were monitored 4 to 8 daily hours, and every 20 minutes through instance scans, we had a sample of which individuals were in the group at a certain time. So this added up to more than 50,000 scan samples, giving us subgroup composition from which we could draw associations understood as the presence of two individuals, the co-occurrence of two individuals in the same subgroup at a certain time point. So altogether, we're analyzing 93 identified individuals in this analysis, including 63 females and 30 males. And yeah, and we used these data to build a temporal multiplex for associations. So again, just to emphasize, we're just considering one type of edge, one type of interaction. Well, it's not an interaction, it's an association, but yeah, one type of link in these layers. And nodes are individuals older than 5 years old. So we stripped out all the infants and the juveniles because they basically move with their mothers. So they're like unavoidably associated to their moms. So their mothers' associations are basically indicators of what they do. Okay, so edges. Okay, so this is a non-directed weight networks, right? And we use the simple ratio association index to construct these networks. Interlayer links are all the same, so we're not making any difference between layers. And these are temporal networks where you can only have links between adjacent layers in time, so they're time consistent. Right. So we're analyzing 4 different scales. And for now, I'm just going to show you some results on the annual scale for the sake of time. And here we see. So this is an example of one of the annual multiplex where each layer represents a year of association data. So the yellow spheres are females, the blue spheres are males. It's 2 different representations on the top and the bottom of the same thing. And time runs towards the left, from left to right. So here we have 75 nodes and more than 3,000 edges. And just to show you the reducibility analysis. So this is case for this network where, okay, so this is the curve that I showed you before in the example. So you can see here how most of this part of the curve is pretty much flat, showing that there's not much difference there isn't much difference in these layers. So if we just look at the highest point, like the highest value, it would suggest that the more optimal partition of this or configuration of this multiplex with respect to the aggregates, it would be made of 20 layers. But I'm not really using this framework to decide how many layers to leave. I'm just actually wanting to use it to see how much variation or redundancy I have through time. So what you can see is I was more interested in looking at sharp changes in the slope of this curve to see if that indicated jumps, big differences in what was going on in associations with through time in this case. And that's sort of the indicator of possible shifts in the social dynamics of associations. And that's how we're kind of looking through this, no? So this is one shift, and that would group all those spots of weights together. And I just want to say that something that came up clearly is that there was temporal consistency. So all the years were grouped together with adjacent years. So we didn't find, and that wasn't something that was prefigurated on the analyses because I let it consider all the layers independently. So that means that you have a progressive change through time, basically. And if we look at the sharpest changes in slope, like here, we can see these 5 blocks of years sort of suggest, which kind of stand out. And as you go further down the curve, then your bigger blocks of time. If we looked at the 3-year multiplex, which I'm actually going to skip for now. Okay, so just looking at the annual example, see, we found temporal consistency in layer similarity, which is what I just mentioned, right? And this suggested that we have more consistency or redundancy between for 2-year to 3-year blocks of association. So what does this tell us? Well, for periods of 2 to 3 years, there doesn't seem to be much change, relative change in what's going on with the associations. But then you get these jumps for 2 to 3 year blocks, which comes up as those jumps in the curve that I showed you. And if we go on a broader scale, we find 2 main blocks of time before 2006 and after 2006. So just to wrap up, this is what sort of thing that I wanted to use the analysis for, to try to find cues of things in the dynamics that might be worth looking into. But what can we what can we do with it? So what, right? What's happening in those years? Why does that curve? What does that mean, right? So right now, what I'm trying to do is explore multiplex descriptors or properties that can allow us to do comparisons between the multiplexes drawn at different time periods on those curves and different characteristics. I'm trying to devise some of them that I've been seen in other papers, but this hasn't been done very much. So, like, finding ways, comparing networks is always messy and it's never trivial. So doing it for multiplex networks, even less so, right? And then finally, I'm just gonna say, because I'm running out of time, I'm sorry for that, that the other thing, and which was probably most interested, is to try to link these big divisions of time, which I'm showing on the bottom here in yellow. Here we have the blocks of time that was the main blocks of time that were detected in the reducibility analysis, to different demographic or ecological features that might be interesting and might give us a sight of what's going on, what's giving rise to these jumps in the curve and in the dynamics as I'm interpreting it. So things like group size or operational sex ratio, or for instance, variability in subgroup size, or things like harsh, like hurricanes, or like abrupt changes in the environment that might be driving changes in what was going on socially. And yeah, well, I'm gonna leave it at that for now. Okay. Thank you, Lauren.
Okay, Sandra, thank you very much for that really interesting talk. I think for the sake of time, I'm going to round of questions and go to the next speaker. But if people have questions later in the fine, we'll have that time after the 3 students have presented. So thanks again, Sandra. So next I'm going to share my screen again, and I want to introduce Nelson. Okay, so our next presenter is Nelson Galvis. Nelson did his undergraduate at the Universidad del Tolima in Colombia, and then a master's degree at Universidad de los Andes in Bogota, Colombia, and he is now a PhD student at the Universidad de los Andes in Bogota, Colombia. Nelson's research has focused on spider monkeys and other primates. His work really focuses on how social structure influences parasite transmission, as you can see from papers that he's worked on with brown spider monkeys and looking at intestinal parasites across various neotropical primates. And he's also done some work using camera traps to study the sort of the behavior and life history of spider monkeys as well. Nelson is finishing his PhD. He talks about writing papers, and he plans to defend his PhD thesis quite soon in October. Best of luck to you at that time, Nelson. And he is interested in looking for a postdoc in 2024. And for that, today, Nelson's going to be giving us a talk entitled Dominance, Parasite Transmissions, and Changes on the Body Weight of Capuchin Monkeys. And with that, I will stop sharing my screen and turn it over to Nelson. And Nelson, please make sure you unmute yourself as well.
So you can see the screen.
I see your slides and the ones on the left side as well. So maybe go full screen. Excellent, looks great.
Okay, so today I'm gonna present part of the results of my project. I call Dominance, Parasite Transmissions, and Changes on Body Weight in Tufted Capuchin Monkeys. This presentation is gonna be divided in three. I'm gonna give you a brief intro about social dynamics in capuchins. I'm gonna talk about dominance and how the body size can influence dominance, and I call this bigger, better. And also I'm gonna talk about the dynamics of parasite transmissions in one group of tufted capuchins. So most of the primates live in groups, and as many of us know that living in groups has some positive consequences, like reducing predation risk, also allows the monopolization of food resources, but also increase the possibilities of having somebody to groom, or somebody to play, or even increase the chances to find love. But also have some negative consequences. For example, if we live in groups, the chances to get more parasites increase, but also we increase the competition for limited resources. If we focus in this last point, this is one aspect that is really important to establish hierarchies in different species, where some individuals they have high ranks, try to monopolize the resources. But if these individuals monopolize the resources, the question behind of this is how these individuals get there in the high rank. So we were thinking what traits enable an individual to become dominant, but not everything is like in pink color. So there's gonna be some cost associated with being dominant. So we were thinking that maybe there is a cost associated with dominance. So, and we evaluate this. So if we focus on the first questions, there are several studies that shows that having a strong relationships helps to create strong bonds with different individuals in the group. And these strong bonds helps also to get in the high ranks. However, another strategy is show power. Showing is an agonistic power. It's like individuals just try to hit or have aggression, display aggressions to another individuals, just to show that they have a lot of power. And this is something that is really interesting, that many of individuals that take that strategy have high body size compared to others. So some works shows that individuals that are big have more chances to get an encounter against a smaller one. However, being dominant have some cost. So it's energetically expensive. So just imagine that somebody that is just spending a lot of time trying to chase another individual, but also some works shows that there is an indirect cost associated with being dominant. So, for example, McIntosh finds that high-ranked individuals have more parasite richness. However, it's important to mention that to have high parasite richness doesn't mean that is a cost, because different studies show that it's an energetical cost, but another it shows that it's not. So based on all that information, we decide to ask ourselves, does body size have an effect on hierarchy position in tufted capuchins? And is there any cause of being dominant? But also, is there a cause of the parasite loss? So To answer that questions, we study a population of Zapajos-Sopela in Colombia, in the east, in a place that we call Los Llanos. We follow a group. We were recording behavioral data from the focal animal and the nearest neighbor from 6 p.m. to 6 p.m. And every 2 weeks, we were collecting fecal samples and we estimate the forest productivity. And once per month, we were measuring the weight of these individuals. As you see in this clip, you are going to see here in the top, we have a hanging scale, and we used some fruits as a bait to try to get at the attention of the individuals. We hold in a display of the scale, so we were able to know what was the weight of each individual in our group. So we were doing this for almost 18 months. And with this whole information, we estimate the parasite richness of every single individual. We estimate the hierarchies for the group. We wanted to know if somebody, how popular were every single individual in the group, using the centrality as a measure. And also we use an approach like you to prove how the body size influence dominance using beta regressions and also general linear mixed models. And what we found, so we found this. So the forest productivity, it doesn't have any effect on the changes on the body weight for almost all individuals in the group. However, Will, one of the youngest individuals, it has a relationship between forest productivity and changes in body weight. And we also estimate the hierarchy for the group. So in this graph, this heat graph, you are going to find the left side, the names of all individuals from our group. On the top, we are going to find the higher rank individual, and in the bottom, we're going to have the lowest rank. And above the diagonal, we're going to see the probability of somebody is winning one encounter to another, and the below is going to be the summation behaviors. So using and combining this information, so we answered our first question: does body size have an effect on hierarchy position in tufted capuchin monkeys? And for us, the answer is yes. So we found a significant relationship between body weight and dominance probability. So we found that high-ranked individuals are the biggest ones in our group. And then we were asking ourselves, well, being dominant is associated with body weight, but why we have to be dominant? What is going to be the consequences of being dominant? So there is several consequences. The first one is social consequence, and if we see Marco, this is the dominant one, and in the right side we're going to find Dana. Dana is the lowest ranking in our group. We can see that Marco received more grooming than the other members of the group. But one of the interesting things is that all the subordinated ones are the individuals that provide more groom to the other individuals. And then we were thinking also with, well, Marco is receiving grooming, but what Marco has to do to keep that rank. So Marco displayed a lot of aggressions compared to the rest of the individuals. But one of the interesting points here is that Negro, that is the beta individual, and Pacho is one of the subadults in the group. The biggest and oldest subadults is also displayed a lot of aggressions. And what Marcos does, he made a lot of aggression to those two individuals just to keep it there in their position and also keeping his status. One of the aspect that is important to highlight is that 82% of the aggression happens in the feeding trees, and it's not a direct way to measure of monopolization of resources, but that is indicates that Marco is not only like trying to keep these individuals in their positions, but is also a way that showing that the market is monopolizing the resources. What is another consequence of being dominant? So here we can see that the dots has different colors, and this color is associated with the sex and the age of the individuals. The black dots are adult females, the orange ones, the subadult males, and the green ones, they are adult males. And the size of the dots is the measure of the centrality. And as we see, the dominant ones, or the dominants have high centrality, but also have high parasite richness. But like I told you before, like parasite richness, it can be a cause, but not always has to be a cause. Sometimes, like some papers shows that having a high diversity of parasites have good benefits for the health of the individuals. So we decide to prove in our case if really the parasites are an energetical cause for the individual. So what we do? So we dig worms. So we dig worm half of the group. So we using ivermectin. We provide the ivermectin. We extract the yolk from the eggs and put the ivermectin. And we gave the eggs to the monkeys. And we're drinking. They are really good opening eggs and drinking everything inside of the egg. And what we found. So in this graph, in the X axis, we have 5 different months, 3 months previous the deworm, and then we are going to have another 2 months after the deworm. We call the post-deworm. Okay, so the individuals that didn't have the treatment, they didn't have any significant difference in changes in the body weight. But the interesting thing here is the individuals that got the treatment, they got the ivermectin, after the first month, most of them lose weight. That is normal. Some of them, they got diarrhea, and then the weight for most of the individuals increased. And this increasing of body weight is significant. So to take away with this really fast talk, we can see that forest productivity doesn't affect the changes in body weight for capuchin monkeys, at least for these populations. And also the dominance has a relationship with the body size. And displaying aggression leads to monopolization of resources. However, and also the parasite richness is related to dominance. So we can say that being dominant has some benefits, like having more resources, but also the spending of energy leads the possibility of have more parasite richness because the immune system decreases with investing too much energy in some aspects. And parasites are energetically expensive for the host. So is bigger better? I think that everyone can take their own conclusion, but for us, we think that bigger is better, at least to be dominant, to keep or get that high hierarchies. And I want to thank to my 2 students, Daniela Rodriguez and Yun Hao, and the members of the field station, John, especially, and my 2 professors, Paulo Stevenson, Andres Link, and all the institutions that provide funds for this project. Thank you.
Thank you, Nelson. We do have a couple of minutes. Can probably take 1 or 2 questions. If you have a question that you want to ask Nelson now, please put a question mark in the chat. And then I will aim to move on to Selena at 11:50. So I see a question from Eduardo, please.
Thank you. I'm Eduardo Fernández Duque, Yale University. Nelson, fantastic setup and data. I'll go straight to the question. Can you comment some more? I'm so jealous of how you've been able to get body mass data from wild primates without any, in a very, very non-invasive manner. So can you comment a little bit? Were you getting that some individuals just loved hanging from the scale? How many individuals were you able to weigh? How frequently? It's a fantastic data set and fantastic solution to a very, very challenging problem, how we get waste from animals.
Well, Eduardo, we spent a lot of time trying to do that. One of the things that we were reading is that at the beginning we wanted to sedate the animals and provide the ivermectin and have the weight, but it's going to be invasive, and also that we are going to provide some stress, and that affects also the body weight. And then one idea came out, just we use a platform and we cover the platform. But every time that they saw a really bright part of the platform, that the monkeys just didn't go inside of the platform. So we decided, well, we saw in these markets that sell meat with the hanging scale, and we said, well, that could be a possibility, and we just buy the device that have the weight. Just we bought that, and we hang mangoes, we hang grapes, and they didn't have any interest on this. One of the things that are really, I forgot to mention, as these individuals are extremely habituated. You can have them around maybe 2 meters away. And once we open a fruit, this fruit is called from the genus Inga, and they were like getting crazy about this and say, wow, it looks like they are interested in this. And just we hang the fruit and then just start going there. But one of the things that we had to change is that we had the device and also the screen next to the device. And every time that it start brightening the numbers, they run away. So we couldn't measure. So we had to plug a cable and have this display far away from them, just to give some confidence from there. And we were able, with that solution, we were able to measure almost every single individual every month. So we were measuring
That's
around
every single individual, pretty much every month. That's how many individuals, how many months? I am
18,
just trying to picture.
18 individuals for 14 months.
All right. I mean,
Yeah.
for those of you who don't work with primates, I doubt it. I don't know how many people have had any body mass data on wild capuchins with this kind of very, very exciting sampling effort. So thanks for sharing. Fantastic.
Thank you for the question, Eduardo.
So I think in the sake of time, Clara, can you save your question until the end? We need to make sure Selena has time when most of us are here.
Absolutely.
Thank you, Clara. I appreciate that. So again, thank you, Nelson, for your talk. And
Thank you.
now I'm going to introduce Selena, our last speaker. And so Selena Carrizo comes from the Universidad Nacional San Luis in Argentina. She did her bachelor's degree there, and now she's a doctoral student in ecophysiology at the Universidad Nacional del Plata. I apologize for my bad Spanish this morning. Not enough caffeine in me. And that actually brings me to a point. I wanted to say I really appreciate these students giving talks in English today. It's quite, I can't imagine doing in Spanish. So I commend them for this. Selena's research is focused on tucutucos, and she's published early in her career 3 papers, at least in various journals. One was sort of a collaboration looking at sort of the diversity of small mammals in a national park in Argentina. But I think her primary paper and collaborative work she's done with people in her lab focuses on sort of the immune responses in the subterranean rodent, one of the tucutucos. And she's been the lead author on a paper in this that was published in JEZ. And Selena is, as I said, she's currently working on her PhD. She's in her 3rd year and ultimately has plans to do some postdoc work. I think her long-term goal from her summary here and also talking to her is to continue to try and understand the ecoimmunology of tucutucos. And she's particularly interested in working with other people to develop laboratory and field-based techniques that can help her to ask the kinds of questions she's interested in. So she's seeking, I think, collaborations with people in other labs. And today, Selena is going to be giving a talk entitled Ecological Challenges Underground, Ecoimmunology of the Subterranean Rodent Tenemus talaram. And with that, I'll turn it over to Selena, and good luck with your presentation.
Thank you, Lauren. Well, my presentation is titled Ecological Challenges Underground, Ecoimmunology of the Subterranean Rodent Tenemus talaram. Regarding my academic background, I study biological science at the National University of San Luis. There I was granted a fellowship to do my undergraduate thesis, in which I explored some aspect of some effects of wildfire on small mammals in San Luis, Argentina. Currently, in 2020, I started my PhD in the National University of Mar del Plata, in the Behavioral and Ecological Physiology Lab. Currently, I have a fellowship to research some aspect of the ecoimmunology of Tenemus talarum, and I recently received 2 awards for my thesis project. In addition, I have just finished a short stay in La Rioja, Argentina, to learn about the use of biologgers in subterranean rodents. That's why currently I am interested to start to use this kind of devices, tools, and technologies that could help me to measure immunity and behavior directly in nature. So, in a natural context, wild animals are exposed constantly to the high selection pressure exerted by pathogens, among other challenges like environmental stressors. As a consequence, hosts have evolved complex physiological mechanisms such as immune responses. Within the immune responses, the acute phase response, or APR, is triggered by a wide variety of pathogens, such as viruses, bacteria, and tissue damage. APR is a fast and complex response involving elements of the immune and neuroendocrine system. And this response is characterized by pronounced physiological and behavioral changes. The former response consists of hypo or hyperthermia, loss of body mass, increase in circulating white blood cells, release of acute phase proteins and glucocorticoids. On the other hand, the sickness behavior involves lethargy, somnolence, decreased locomotor activity, anorexia, and postures to reduce heat loss and conserve energy to fight against the infection. So this set of physiological and behavioral responses, known as sickness syndrome, ensure the host recovery in a short period of time, but they are also costly in terms of energy, nutrients, time, and opportunities. Therefore, it has been proposed that the APR could be an important mediator of trade-offs between immunity and other life history traits. Although there is a broad consensus about the APR symptomatology, the magnitude and intensity of immune responses vary markedly between species, populations, individuals, and sexes, and have been proposed to be a consequence of life history traits and different environmental conditions. This variation in immune responses in an ecological and evolutionary context is the object of a study of ecoimmunology. Our species of interest is Tuco de los Talas, or Tenemus talarum. It is a subterranean rodent that lives solitary in burrow systems and maintains exclusive territories. So, thinking back in Eileen's talk, we are here at the extreme end of this gradient of social structures for Tenemus, as she mentioned 2 weeks ago. Tenomystilarum is distributed in coastal grasslands and dunes of Buenos Aires in Argentina. The richness and abundance of the parasitic fauna that host Tenomystilarum is relatively low, but the prevalence of these infections is high. So far, what do we know about the ecomonology of tucotucos? Previous research has established that the inflammatory response is not associated with a significant increase in the oxygen consumption, but negative effects have been identified. Sorry, negative effect of restrictive diet and parasites have been identified. In contrast, the adaptive immune response represents a considerable energetic cost comparable to the cost of lactation for the females of this species. Therefore, first we explore the effect of LPS, or lipopolysaccharide, which is a non-pathogenic antigen, a component of the cell wall negative bacteria, gram-negative bacteria. This antigen allows us to activate the APR without the negative effects of bacterial proliferation inside the host. Thus, some of our research questions were: how does the acute phase response develop in tucotucos? Is this immune response energetically costly for this species? In addition, we are interested in the effects of different factors that in their natural ambient could affect the development of APR. Thus, the effect of infection intensity, ambient temperature, and resource availability on APR were evaluated. To explore our questions, adults of Tenomystilarum were captured in Buenos Aires, specifically in Mar Azul, at different stages of this research, then were transported to the lab, and when we finished the experiment, the individuals were released at the same capture sites. In the first aim, we did 2 experimental groups: control injected with saline solution, and LPS injected with 1 milligram of antigen per kilogram of body mass. We measure core body temperature, body mass, metabolic rates before and after the exposures, sickness behavior, different parameters of sickness behavior, such as locomotor activity, resting, among others. In the second experiment, we performed the exposures in different groups of individuals with a dose gradient of LPS and saline solution as a control. And in addition, we measure white blood cells, glucose level, cortisol levels, albumin, triglycerides, among other parameters. As I said earlier, we also measure the effect of ambient temperature and resource availability using a single dose of LPS. But here I'm going to talk only about the result of LPS effect and infection intensity. So regarding the physiological aspect of acute phase response in Tenomystilarum, we found fever at the first hour post-injection with an increase of 1.5 to 2 degrees Celsius in body temperature, loss of body mass in males up at 12 hours post-injection, and a 14% of increase in the resting metabolic rate. The behavioral aspect of APR involves a significant increase in resting behavior, and individuals also spend more time with their eyes closed, totally or partially closed. They also present wet abdomen and depressed hips. Regarding the effects of infection intensity, we found fever without difference between antigen doses, and We also found increases in the metabolic rate at the first hour post-injection that were associated with increases in body temperature. Even in the low LPS dose, we found increases in the number of neutrophils and decreases in the number of lymphocytes. And also we found increases in the cortisol levels that were dependent on antigen doses. To summarize, the physiological aspect of the acute phase response activated by LPS in tuco-tuco de las talas consists of increments in body temperature associated with increases in the metabolic rate that represent an increase between 0.84 to 0.87% of the daily energetic expenditure for this species. We also found loss of body mass in males at 12 hours post-injection, redistribution of immune cells, neutrophilia and lymphopenia, an increase in cortisol levels that were dependent on LPS doses, possibly to enhance the immune function. Regarding the behavioral aspect, we have identified some evidence of associated sickness behavior. Individuals exposed to LPS spend more time resting and with their eyes closed. In addition, during the APR, a high proportion of individuals shown wet abdomen and in contact with the floor, possibly to lose heat by conduction. However, we have recorded other data in different aspects of APR, which are under analysis. So we have record of anorexia, food consumed, different parameters of nutritional state, locomotor activity, resting behavior also in relation to the other aims of my thesis. So the physical conditions of the barrel system, finally, life underground has distinctive physical and ecological characteristics that exert environmental pressures on individuals, which in turn lead to physiological specializations. The physical conditions of barrel systems, such as high humidity, low ventilation, and protection against UV light, in addition to their coprophagic habit, could favor high levels of parasitic transmission. As well as this type of habitat could increase the exposure to soil-borne fungi and bacteria, but decrease the exposure to iron viruses. On the other hand, the solitary habit could decrease the exposure to parasites. Portenomys stylatum, and we could think that this also happens for other subterranean species. This immune strategy against extracellular pathogens could be the result of immunity evolution in this environment in which this rodent lives. So thank you for your attention, and thank you to my advisors, Ana Paula Cutrell y Roxana Cento.
Thank you, Selena. Very interesting work there. So we have a little bit of time if anyone wants to ask questions directed towards Selena first. I can take one or two questions to her, and then
I have a question mark there.
Yes, go ahead, Clara, please.
Yes, on Clara B. Jones' retired social evolution. I'm going to limit my question to Sandra. Clearly, I do not know network analysis, and I don't know very well your study, but it occurred to me in your temporal analysis that you could ask a broader range of questions in addition to your network analysis if you use time series analysis on top of what you're doing. For example, you could study across time various contexts and various, for example, group size or subgroup size, or various components of your phenomena, given different feeding sources, different fruiting trees, just a huge range of questions with time series analysis. What do you think about that?
Thank you very much, Clara, for the suggestion. Well, I mean, I think that's one option. And right now we're precisely kind of looking at different ways of tackling the dynamics, the questions on dynamics. And today I focused on the multiplex framework because I kind of started trying through a methods exploration just because it is part of the focus of the project to see the potential of that framework. But I'm very interested in comparisons between what we can learn from different methodological frameworks for the questions of what's driving the dynamics. And so far, the fact that the things that are the signals, if you may, that are being pointed out by the analysis I did on places where there may be shifts or significant shifts in the changes in the dynamics, I think are really good places to look with other tools at before and after things and different approaches like the ones you mentioned. So for sure, time series analysis is one possibility, and I appreciate the suggestion. Thank you very much.
Thank you.
Okay, thanks, Clara, for the question. Just to remind everyone, please, if you have a question, post a question mark in the chat, and I will call on you in the order in which the questions come in. And please tell us your name and where you are, so we can get to know each other. And again, we really try to encourage students to participate. So please ask questions. And for the students in the crowd, and you're worried that you don't have a good question, well, there's a history here of me having a question in my head and other people ask it. So it gives me more confidence that I have good questions. So please ask questions. We want you to be participate. And if you're on YouTube, Logan is watching. He'll relay your question to the community. Please post a full question in the YouTube chat. Okay, so our next question goes to Nicola.
Hi, everybody. I'm Nicola. I'm studying multi-layer social networks of Arabian bubblers. I'm studying in Israel. So because there is not a lot of birds with multi-layer networks, I mostly read primate papers. So thanks to Nelson and Selena, their talks were very interesting, but I really have to ask Sandra a few questions because I don't have a lot of multi-layer network people. So I have a lot of questions, but the most basic one would be in your first graph, you had different layers, and in each of them, individuals were connected inside each layer with different interactions, I assume. And then you had the connections between different layers. So we already have a lot of different metrics like centrality, degree, betweenness, and so on for each layer. What are the metrics for connection between layers? And is there just one? Is there multiple? And how we calculate them? Because for me, that was by far the most challenging part of multi-layer networks.
Hello, Nicola. Nice to meet you. Thank you for the question. Well, that's not a trivial question for me either. I mean, dealing with intra-layer edges is already messy. Dealing with interlayer links makes it more complicated. And so far, I've only worked with like unit values, like all the interlayer links are the same, but that's an area of that could be very interesting in the future to play around, especially for interaction multiplexes, not in the temporal case maybe, but yes, when you have different interactions, I think using different probabilities based on the occurrence of behaviors or such could be interesting. But anyway, in terms of metrics or multiplex properties at the level of the multiplex, that's one of the things that's keeping me busy. And well, there's some metrics, and I have to say that I didn't mention it. I'm sorry. I've been mostly using MoxVis, which is in R, which is developed by the Domenico. So I've kind of stayed on tune on the metrics or the possibilities that have been provided by MoxVis. But I've also been trying to think of other things that would make sense, especially across different multiplexes. So I mean, a basic one that he uses, which depending on the type of group you have can be very or non-informative at all, is the amount of edge overlapping. So like global edge overlapping, it looks at how many of the edges that you have in one layer occur throughout all the layers. For example, what proportion of the links you have are prevalent throughout the whole thing. And you also have extrapolations to the giant component idea. So the largest connected component of the network, but in different ways, considering intersections like throughout the different components of the layers. I mean, at least MoxVis offers, or there's three conceptually different ways to think about the largest components proposed by the Domenico. There might be others, but those are the three ones that he has proposed. In his late, I don't know, maybe you've seen it, the book he published in 2022. It's been very helpful for me to understand a bit better some of the things they're proposing from that side of things. And other things that I'm looking at a lot are coefficients of variation of things like density, like throughout the layers, and coefficient of variation of transitivity, of clustering. I mean, just the variation rather than the values, because I'm interested in maybe being able to compare between different networks. So I just want to know how much variation I have. I think those could be, could maybe be useful, but it will depend on your question and what you're looking at. So for example, with a very densely connected group, some of these get very boring because you almost always have a fully connected network. So then the density won't tell you much, no? For example. I hope
thank
this.
you very much.
Yeah, you're welcome.
Okay, thank you. And thanks for that question, Nicola. If you have more, please put a question mark back in the chat and we can continue. The next question is to Anne-Marie.
Yeah. Thank you. I'm Ana Marie. I'm a postdoc at Universidad Católica in Chile. And great talks for everyone. But my question is directed to Nelson's talk, because I was very fascinated by like the plot that you have with grooming received and given, that some of the monkeys actually didn't receive any grooming or didn't provide grooming. So I don't know if you can kind of like, do you know what's happening there? Does it depend on like kind of their social organization?
Thank you for the question. To be honest, I don't know what is going on there. That is the real answer for this, because many times when you are following monkeys, when you are following a species that have so many individuals, you are not able to see the whole individuals all the time. For that reason, we spend a lot of time in the field for so many months, but even spending a lot of time, 12 hours per day for many months, we didn't see any interaction from them. So that is it. So sometimes we were wondering why these individuals, and if they don't have any bonding with another individuals, but they play with others. So it doesn't mean that, well, they have another social interaction, but not grooming. And if they are doing grooming, we didn't see really often, or we didn't see because maybe they are not really frequent.
Thank you. Yeah, and then it also brings me to another question. Like, I don't know which class of parasites you looked, if you only looked at endoparasite or also
endoparasites, just endoparasites.
Oh, okay, because then I can, if it was also ectoparasite, I can imagine that the ones that are grooming the most dominant will also receive the ectoparasite of the most dominant. so that even the richness, I don't know if you have the opportunity to also look at ectoparasites, or
No,
that's just like outside of, no, okay.
no, because to see that we have to take the animals, and we don't like too much the invasive method.
Well, thank you very much.
Thank you for the questions.
Thank you. I'm next in the queue, so I'm going to ask a question. This is directed to Selena. By the way, thank you all for your great talks. Really fascinating stuff and encouraging to see really great work coming out of your labs and your projects. So Selena, there were some pretty significant body mass losses, you know, like upwards of 10–15%. That to me seemed like a lot. And even in the controls, there was some body mass loss. So I guess I had two questions, and maybe there are things just to think about is, you know, where do we start seeing significant fitness costs with that kind of body loss? Do you know about that in the field, or like in terms of survival or reproduction?
Well, thank you for the question. No, I don't have an answer for this question. The idea of my thesis is to start work in the field, so we are generating information now from captivity, so I don't know what happened in the field yet.
Okay.
With the field.
And thank you. And so I have a follow-up question, if it's okay for you, Selena.
Yeah.
And I apologize if I missed this in your talk. Do you have any? It looked like the females, there was no significant difference between the control and the experiments for the females, but there was one for the males. Am I correct in that?
Yes, for the body mass loss.
Yeah. Yes, I'm sorry. Yeah, for the body mass.
Yes.
Do you have any? And again, I apologize if I missed your comment on this in the talk. Do you have any idea why males are so much worse off than the females?
I don't know the reason why, but always the males lost more body mass. They present more increases in body temperature, but I don't know why this happened. But always they also present more sickness behavior, and the females are better coping with infections, but I don't know the reason.
Thank you. Be really interesting trend there, I think. Okay. So Eduardo, I'm going to skip to Baptiste and then I'll come back to you. Okay. So the next question is from Baptiste, please.
Hello, everyone. I hope you can hear me okay. I'm a PhD student at the University of Göttingen in Julia Ostner lab. I had a question for Nelson. Thanks very much to the three of you for the talk. Nelson, maybe I think I missed, and you probably said it, but I was super interested in your relationship between body mass, parasite richness, your ivermectin treatment, and then you link it to dominance. And it felt to me, I think I missed one link, that you found the relationship between parasite richness and dominance, also dominance and body weight. And then after the ivermectin treatment, you saw an increase in body mass. And you eventually interpreted as evidence for a cost of parasite richness. But I was wondering, did you look at parasite load, some form of egg mass per gram feces or something? Because it felt to me like the ivermectin treatment might really lower your load. Also maybe influence the richness, but I was maybe missing the link, and I was wondering, did you look in the background whether parasite richness in your study was linked to parasite load or something like this?
Yeah. So one of the things that because of the time, I didn't mention, but I was, after the treatment, I was looking for the parasites, and I was sure that those individuals that got the treatment, they didn't have any parasites. But after the 3 months, the third month after the treatment, they got again parasites. And I don't know, it looks that it's something natural, that because of the interaction with another individuals, because they go to the ground, they got again parasites. So for that reason, I just stopped measuring just until the second month. That is one of the things. And one of the things that I forgot to mention, I didn't forget, I didn't include, and it's just because we have a treatment for just 6-7 individuals, but we compare the ratio of growth for the different individuals, for these that got the treatment and the other that didn't have the treatment. And the interesting thing is that the individuals, they are dominant. After getting the treatment, the rate of growth is bigger than the other individuals. However, when we pass to another window of time, we did the same without getting any treatment. All these, the high-ranked individuals, the rate of growth is lower than the other individuals in the group. So in some way, we can't say directly that it's a cause being dominant, but we can say that the parasite is an energetical cause for those individuals. That is indirectly that our reading of the situation. And I don't know if I answered your questions.
Yeah, completely. I think your last results that you were just mentioning are super interesting. So yeah, if one day you present them again, or if they're in the paper, I'm super keen on seeing them. Thanks.
You're welcome.
Thanks, Baptiste. Our next question is from Eduardo.
It's always very exciting to hear from our young colleagues. I'll ask the question specifically for Sandra, but I think it also applies to some of the things that definitely Nelson could ponder. So as we continue gathering multi-year, multi-decade data sets, I was wondering, as I was listening to you, first, thanks for emphasizing methods. You've already heard me say that we need more of that, since that's what defines what we do, methods. Have you considered, are there ways in which you can try to predict, really split your data set in different ways and use some of the data to train your model, etc., and see how well it predicts the other? Or more exciting even, imagine any of you guys pre-registering some future analysis to be done when you collect data on social networks during the next 4 or 5 years, and see the extent to which your ideas and your understanding of past data manages to anticipate what's going to happen. Because that's been the history of what we do. We always look back and try to make sense of what happened, but probably one of the most robust tests of us having understood the system is being able to predict what's going to happen.
Thank you very much, Eduardo. Well, I'm lucky enough to be part of this big group that I showed you at the beginning of my presentation, which includes 2 physicists, and they are actually working on a mechanistic model to try for bond formation for associations and other interactions that we're working on with the monkeys. So what we're doing right now is, one of the things we're doing is playing around with their model, the layers generated, association layers generated by their model based on the data. Their model was trained, or it was based on part of this data set. And to try to create layers and build multiplex networks from temporal association multiplexes like the ones we have, or I showed you, and try to see how they relate in their properties. And so that's one of the reasons why I'm curious or interested in looking and pinpointing, useful metrics or properties at the multiplex level to see how well the model or how unwell the model reflects upon the actual real network or empirical data. I mean, they are working on how to scale up their model to a multi-layer framework, which will be really exciting. For now, we're doing it this other way, where we're kind of using the monolayer mechanistic model to try to build on associations in a similar sense. So that's the idea. Well, I mean, we're trying to, I think, touch on a bit on what you're saying through that method. Because they identify certain processes for bond formation that could allow us to explore if they are playing an important role in the formation and the dynamics of the associations. But yes, I mean, we have that in mind. So to try to look at the predictive part, we haven't thought about maybe using the first half of this data set to try to predict the second half. That's, I didn't have it in mind. So thank you for the suggestion.
I'm not an expert. I'm far from being an expert on this. I was not suggesting using the first half. Actually, what I think is you could, that's where we need advice from more quantitative oriented analysts, but more like an approach where you can do some resampling and produce a sample out of the total and use it. So not necessarily the moment you just take the first half and the second half, you're already introducing a lot of confounding in the two samples that you're arbitrarily deciding. But there are people who can probably help us figure out how to do it.
Yes, I oversimplified. So yeah, taking a sample and subsample to try to model or scale up or predict and compare. So yeah, thank you for that.
Thank you.
Okay, I think our next question comes from Adriana.
Hello, I'm here from the count from Adriana, and we are from the ecology behavior ecology class. And my name is David, and I have a question for Nelson. That is, during the grooming, can there be a detection of a considerable amount of parasites? And in the event that they are noticed, could it cause a distancing or a social break between the individuals?
I'm sorry, can you repeat the question? I didn't understood.
During the grooming, can there be a detection of a considerable amount of parasites? And in the event that they are noticed, could it cause a distancing or a social break between individuals?
Okay, and related with the first questions, we saw that grooming, well, there are so many benefits called from grooming, like creates strong bonds between individuals, and one of the others is that it helps to take out parasites. But another papers shows that the action of groom also is a way to get parasites. And I have a paper about it, and the grooming is not really a good thing for the individuals who does the groom. It's a way to get the parasite from the other as well. And respect to the second question, I didn't evaluate that. I'm not sure, but I saw that some paper from Colin Chapman that he found that, or he suggests that individuals with infections, high infection of parasite, sometimes they are taken away from the group, or individuals don't interact too much with them. But that is something that is the only paper that I have read about it. I don't know, maybe I can check the data and do some social network analysis using this and see. Thank you for the questions.
No, thank you for the answer. Thank you.
Okay, I don't see any more questions in the chat. If anyone has additional questions, please feel free to add one to the chat. I see one from Filippo. Please go ahead.
Yeah, I am Filippo Relli from
Filippo, it's very hard to hear you. I'm sorry to cut you off.
Can you hear me better now?
Yes, very much so.
Okay, yeah. I'm Filippo Aureli from Universidad Veracruzana in Mexico. And I have a question for Nelson. It's a follow-up question about the first question Eduardo asked you about the way to get access to the animals to have their weight. And I think I'm impressed, as Eduardo was, of the system, but especially looking at the dominance, you get the weight for everybody, since you are putting a resource that certainly the animal likes, because as you explained to us, it was not easy to find. There wasn't any problem in getting everybody, and not that the dominance or some individual they are more dominant than others were excluding some individuals to do it. So just again a more technical question, but out of curiosity, because it looked like looking at dominance. So and then you managed to get everybody. Seems impressive.
Yeah, one of the things in the beginning is well, how is the whole process? We hang the scale, and we hang the bait, and normally the dominants they take the initiative to go there first. So alpha and beta were all the first ones, but we were working in a group with my other students, so we have to keep the dominants busy. So we have to show them the fruits and throw it away. So in that way, the other individuals were able to go and grab the fruits. Otherwise, they are gonna keep all the resources for them. They're monopolized the resources. So that was the way. And after a while, just the dominants just get tired, and maybe they were completely full, so they didn't pay more attention to the scales and the fruits hanging from there. So, and then maybe the floatants, I don't know how, the Ateles individuals of the group, they decide to go there, but normally they were always the last one. So many times we were frustrated because we were thinking that we are not gonna get away from them, but after maybe half an hour that everybody went, just the satellites went and took the initiative, because they are more cautious to go there. So
thank you. You made it very easy when you told us, that then everybody got it every month. I felt like, what? So now you explain a little bit about effort. Yeah. Thank you.
It talks a lot of time. And in the beginning, I was saying that with Sandra and Adriana Maldonado, we were talking about it. And in the beginning, we wanted to decide to work with woolly monkeys. We decided to do the whole system with the woolly monkeys, but the woolly monkeys, for them that don't know about how they behave, they are boreals. So we hang a platform. So we were pulling the platform high up in the trees, and sometimes they go there, but sometimes didn't. And most of the time the individuals hang from the tail. So many times we weren't able to weight properly them. So we decided to work with these top-depth capuchins also, because they were habituated. We were working with them for a couple of months. So, and then after several intense of waiting, it works. Thank you, Filippo, for the question.
Okay, so we have another question. This comes from, I think, Adriana's class, Camilo.
Hi, my name is Camilo Arenas, and I'm from La Universidad Rosario in Colombia. So Nelson, from one of the articles you suggested, I have the following question, and it is: in a society in which a high-rank female is groomed the most by other males, and the interaction with males is close to known, would we expect males of high to low class to be less susceptible to infection, since this exclusion from grooming may avoid the vectors to spread to these social groups?
Well, I I think that I know what is the paper that you are talking about. And I think it's the Macintosh. And basically, there are several explanations for this. And one of the main results that from many people that work with dominance and social networks is that dominance, the tendency is that the dominance always is going to be the central individual. It means that they are going to have more interactions with another individuals in the group. But being also popular in a group increase the chances to get parasites. So those individuals that have less interactions have less chances to get them just because they are not exposed to this. I don't know if I'm answering your question.
Yes, but it is also related like to sex, because in the article they said male.
It depends the species. It depends on the species. In the article that I mentioned, the females are the dominant ones in this. So in this case, for us in our populations, the males are the dominant ones. So it depends on the species.
Okay, thank you.
You're welcome.
Okay, I don't see any questions from YouTube, and I don't see any more in the chat. If there's any more, please post them now. I guess I have one question that might be applicable to everyone, and this is more of a, I don't know, give you an opportunity to sell your organism. If there was one thing about your study organism, what do you think is most important about it? Why is it such a cool study organism? Sorry, I didn't mean this to be a comprehensive question, but I just think it's a really important question that we all have to think about from time to time.
Well, should Nelson answer for the parasites for which of all the species of monkeys he works with? And the plants. Well, I'll say about spider monkeys just to follow on the question. There, I mean, not only the fission fusion thing, but they're just so I don't know, they have all these behaviors that are enigmatic to me. Like, I mean, they hug, and they, at least the group that I studied, they seem to even, I mean, they do this like use of plants to males to hype themselves up. They do raids. I mean, they have all these chimp-like things as well. Yeah, to me, they have so many complex different, I mean, their ethogram is huge, and that just keeps, at least for the year and a half that I was lucky enough to be working around with them, there was always something new happening. And I'm sure that happens probably to everybody studying animals, but they were like surprisingly unexpected things that would continuously come up in terms of their behavior that, yeah, that were very surprising to me.
I agree with Sandra. I think that I started working with the spider monkeys, so for me were fascinated, but not just because the fission fusion, as Sandra said. It's also, I believe that they're extremely smart to know how are the distribution of the fruits in the forest, but also knowing when is the time to go to look for them. I think that this is a kind of things of memory. I think that is one of the things that I really am interested on. But after I start working with capuchins, I think that capuchins are amazing, are like unbelievable characters. And I work with also white-faced capuchins, and for me it was really surprising to see them just taking fruits from the tree and just hanging below some dry leaf, and after 1 week later they go to the same spot and check for this fruit and be able to open a hard fruit that the only way to open it is get fermented. That was the only way, and that for me was extremely surprising, and that for me is extremely cool. And I think the cappuccins for me are cultivating my interest a lot.
Well, for me, I think that, well, they are cute, obviously. But, well, the most of research there are about immunity are with laboratory rodents. So we are working with the non-model species that are subterranean, that are solitary. So and also they present a lot of high variation between individuals. So it's difficult to understand, but it's so interesting also.
Those are all really good reasons. We have another question. Alba, please go ahead.
Hi everyone. I'm sorry I missed the first two talks because I was really confused by Argentinian time today. So I listened to your talk, Selena, and I have a question. I was actually trying to Google this information, but I should ask an expert. So I think I read something about the mating system of tuco-tucos recently that I found like very interesting, but I don't remember what it was. So can you tell us a little bit more about their mating system? Like you mentioned, they were solitaries, but how do they reproduce or mate?
Well, they are subterranean. They only go outside for food, and they are solitary, and well, they meet for copulate. Obviously, they are polygenic. I don't know how it's
Polygynous. Polygynous. Okay.
I don't know what.
Do you know if is there female mate choice kind of thing in the species? I think I read something about like assortative mating in females or something like that.
Yes, I am working in the lab with other partners that are working in this topic, a specific topic. But there are chosen. The females chose
the males.
Sí.
Okay. Well, thank you.
Well, I think it's about 1:15, and the three students have meetings after this. So at this point, I want to, before I shut down the YouTube, I want to thank everyone, Sandra, Nelson, Selena. Thank you for really engaging, exciting talks, and for participating in this today, and for everyone in the Fine here for engaging with these students. It's been a pleasure hosting you, and thanks again for being here.
Thank you, everybody.
Yeah, thank you for everyone, to everyone, and thank you for the space. I think it's important to show what we do here in Latin America as well.
Thank you for the opportunity.
And thanks to Adriana's class for participating. It's great to see the students engaged, and I hope you guys show up again to more Fines. And with that, I'm gonna shut down the live stream, but people